I want to join you. Okay, so let's finish up this chapter. Everybody understands? What's this? What's this? This is DNA, and what's happening in this figure? We're duplicating DNA. Why are we duplicating DNA? Because we were just finishing up our discussion about mitosis. In order for me to make two brand new cells, two daughter cells, in the mitotic stage of my life as a cell, I have to replicate my genetic material so both those brand new cells has the map. I keep calling it the map. The map for what? This is your map. This is why you look and act and sound the way you do, believe it or not. Yes? Because of your map. So, how do we read the map? What does the map mean? You all have a map, right? Does anybody have a map? Most all of us have a map of some kind somewhere, right? Got one shoved in the glove compartment, a big one in Maine. If you're really cool, you have one of those books. The Gazetteer, right? Is that how you pronounce it? The Gazetteer. Do you all use every single page of the Gazetteer every day? But you have the whole map, right? Every single cell in your body has the whole map, for the most part. But might not use the whole map every day. Maybe I just need to go from here to Waterville. So I'm going to flip to that page and use the map to go to Waterville. Yes? Maybe I'm a cell that needs to make a certain chemical for you. I need to use the map for directions for everything that I do. And the directions that I'm talking about is what we start talking about under protein synthesis. So we've already discussed a cell's life. S phase. I hang out, I get bigger after I'm born, I do what I need to do for a living. What's that called? Interphase. Then when I go to the S phase of interphase, I've decided to do what? Replicate. So I have to make a copy of all my genetic material, right? Because if I'm going to move on to two brand new cells, everybody has to have the same info. And then when I start the mitotic stages of my life, I'm now going to take the copied info and make sure both my brand new cells get it. What are the stages of mitosis? Prophase. That's when I pack everything up. Make sure that I get it all tucked away nicely, ready for the move. Metaphase. I'm going to line everybody up. All that replicated genetic material in the middle of the cell. And hook on one of my little fishing poles so that I can then go to anaphase, where I'm going to do what? Separate the copies and pull them to opposite poles. Then what else does my new cell need besides just genetic material? Yeah, it needs all the other stuff we talked about when we talked about cells. It needs a mitochondria, it needs endoplasmic reticulum, and all of that good stuff. So at the end of anaphase and through telophase, another process called cytokinesis is going to happen as well. Right? Everybody with me? What's cytokinesis? Replication of what? The organelles in the cytoplasm. Remember, there's organelles in the cytoplasm. So if I ask you that question, don't just say cytoplasm. Understand that it's all of the organelles that are replicating in the cytoplasm during cytokinesis. End of anaphase and through telophase. In telophase, I'm going to set up my new house. Those two new cells are going to reorganize, regain that nuclear membrane, make sure that both of them are pinched off with their own new plasma membranes, and that's going to happen at telophase. Yes?
Online we had for our lab the last question of this past week's homework said something like, at what point does the chromosome or something have one strand of DNA, at which of the 4 phases? And I couldn't find it anywhere. I happened to guess the right answer, which was, I think, telophase and anaphase or something.
That question, one strand, you mean a single copy of genetic material?
Well, that's what I didn't understand what it was saying.
Okay.
It was
Should,
unclear. I did make a little note to Pearson that
well,
I didn't like
talk to your lab instructor about that. Peer, yeah. They're the ones that chose the question to put in your,
oh,
so you should talk to the lab instructor about that.
So in, when you see, because like you asked the question and I said cytokinesis, but it was actually telophase when, that's when they start to form, to split, but cytokinesis isn't actually when they do.
No, cytokinesis is happening during telophase and at the end of anaphase.
Okay.
So cytokinesis is the replication of all the organelles, and that's happening during the end of anaphase and during telophase.
Well, okay.
Telophase is when the two actually pinch off to create two separate cells.
Because in the book it shows it after telophase.
Yeah, because you're looking at the pictures and not listening to the explanation.
No,
Yeah, but that's what I'm saying. So it's not, it's misleading sometimes, the little pictures. But what I'm saying is, remember, it's happening during the end of anaphase and during telophase. And understand what it is, because it makes sense. If I'm going to make 2 brand new cells, I have to have all the organelles as well. Okay? And actually, I think in the description in your book, it's under telophase too. But it happens after. And if you look at that picture closely, let's go back and have a look.
it shows the whole.
Look at the little boot. Yeah, and that's what the boot means. So it starts here and is going to continue all the way through telophase. Okay? Questions?
The book does say in several places that cytokinesis is the division of the cytoplasm.
Correct.
Cytoplasm meaning,
What's the cytoplasm? What's in the cytoplasm?
does that include organelles?
Correct.
Okay.
If you just want to talk about the fluid, you call it cytosol. If you're talking about the organelles that are floating around in that fluid, you call it the cytoplasm. So that's why, again, that's the definition of it, but understand, what I'm trying to say to you is understand the definition. Understand what that means. Do you ever go to the dictionary and look up a word and it uses words that you don't understand that doesn't mean you now understand the definition of the word, right? So you have, exactly, you have to go and look it up and clarify. So know the stages of mitosis. Next in your book, we are going to talk about protein synthesis. And this is how I use the map, what the map is for. What is the map made of? What's the map made of?
RNA.
It's made of DNA. What's DNA made of? It's a nucleic acid made of what? Nucleotides. Nucleotides. How many different nucleotides does DNA have? 4. A phosphate, a sugar, and nitrogenous base. Adenine, guanine, thymine, cytosine. That map is a sequence, a very specific sequence of those 4 nucleotides in a very specific order. That's your map. Now, how do I read the map? And that's what they talk about under transcription and translation. So when I look at DNA, the molecule, I see a double-stranded molecule, nucleic acid, that has a very specific sequence of these nucleotides. Is my DNA the same as your DNA? No. Actually, believe it or not, most of it is. That's what makes you and I both the same species, Homo sapiens. But there's enough of it that's very different, and that's unique to me. But I have a lot of similarities to you as far as the types of proteins I make, the types of cells I make, the types of organs that are formed from those cells. That's where we get the information from, our genetic information. But we have to be able to translate and transcribe that information so that I get the result, the proteins that that information is going to tell me to make. So that's what we talk about when we talk about transcription and translation. Now, DNA is a huge molecule, right? We just looked at it. It's enormous. It's so big that it doesn't leave the nucleus. But if I need directions someplace else in the cell as to what I need to make and put together, how is that information going to leave the nucleus and get there? I need some help. I'm going to rely on another nucleic acid to help me out, to get the information that's contained in the DNA and then go actually make the protein that the DNA wants me to make. My helper molecule is also a nucleic acid. It's called messenger RNA. Messenger RNA is a single-stranded nucleic acid. What's it made of? Say it loud. Nucleotides. Except there's one little difference. RNA has one different nucleotide than, yeah.
It was single-stranded?
Yep, single-stranded. Okay? This is going to help me get the message out. Take the information from the DNA and then go produce a protein outside the cell. Outside the nucleus, excuse me. Messenger RNA is made up of 4 nucleotides, just like DNA, except the 4 nucleotides are a little bit different. I have adenine, just like DNA does. I have guanine, just like DNA. I have cytosine, just like DNA, except I don't have thymine. Instead, the complementary base pair to adenine is going to be uracil. What does she mean by complementary base pair? Basically, what I'm going to do inside the nucleus during the transcription process of protein synthesis is I'm going to create a copy of DNA. And I'm going to do that by actually matching up base pairs in an unzipped portion of DNA and making sort of a copy of the code with messenger RNA. Now remember when I replicated my genetic material back there in mitosis? And I had all these little free nucleotides running around inside the cytoplasm of the nucleus? That's how I made my copy of DNA. Who is going to help me do that? Nope. An enzyme, right? What was the name of the enzyme that was going to help me make a copy of DNA? It's called DNA polymerase enzyme. I got other enzymes. This enzyme called what? Now it's a ligase enzyme. It's a polymerase enzyme too. It's called RNA polymerase. It's going to help me make a copy of the information. The information comes in the sequence of nitrogenous bases. I'm going to make a complementary strand of that. So every time on my DNA that I unzip, I see an A, what's going to come and attach to the A? No. I'm messenger RNA. I have no T. A U. Every time I see a C, what's going to come in? G. You getting the point? And I'm going to copy that information. That's the translation part of transcription and translation. Excuse me, transcription part. It's kind of like the big boss can't leave the office, yes? So the secretary has to come in and take down exactly what the big boss says. That's the messenger RNA formation. So that information is being what? Transcribed from the DNA. Who's going to help me do that? Enzymes. The next step, I'm going to make that big long strand. Remember, I have the whole map in every single cell. So I have the map in my genetic material, my DNA, to make every single protein my entire body would ever need. But if I'm just going to Waterville, am I going to look at the whole map? No, I'm just going to look at that page of the map, right? If I'm just making insulin, do I need all of my genetic material to do that? What do I need? Just that portion. Just that portion of the code that's going to result in insulin, that particular protein. So I only have to unzip a certain portion of my genetic material during transcription and translation. So I'm going to then read that whole portion of the map that I need. And at some point, I don't need any more information. I'm done. I know how to get to Waterville. I'm good. I go to Waterville. What do you do with your map when you're done and you know where you're going? Or you got there? You close it back up and you put it away. Or you terminate. Yes? So we also need a termination sequence. Directions in our genetic material that are going to help us read the information. So during the transcription process, and we see this on page 103, we have the initiation. Unzip the DNA and start the process of creating an RNA. I'm reading the code. Then I elongate, then I terminate. Who's going to help me make that messenger RNA strand? Who's going to help me make it? Who helped me make new DNA? Enzymes. What kind of enzymes? Polymerase enzymes. So RNA polymerase enzyme is going to help with the process of reading the code, matching up those little RNA nucleotides to create a strand of messenger RNA, a copy of information from genetic material. Once I've created the strand of messenger RNA, in the code itself, there's a lot of extras, extra information. Just exactly like when you read a map. There's a lot of stuff on there that you might not need. You know, it tells you where all the hospitals are, and it tells you where all the parks are. I'm not really going there. I'm just going to Cold Stone Creamery, so I'm good, right? So another part of this process is to sort of snip out any extra little bits. So the messenger RNA, once it's created, has to be processed. All right, now I have a strand of nucleotides, a mirror image, a copy of what I found in the genetic material. I'm going to take that copy and I'm going to bring it outside the nucleus. So messenger RNA is going to leave the nucleus and it's going to go park itself in a place that's going to help it create a protein, a place where translation can take place. Does anybody know where I build my proteins inside cells? On the ribosomes. Ribosomes are also made of RNA. It's called ribosomal RNA. And there's two bunches of RNA that act sort of like a little knitting machine for all proteins that are made. That messenger RNA has a little place where it can dock itself. And now, with the help of another RNA, do you know what the other RNA is that's going to help us? Transfer RNA. I'm going to begin to build myself a protein. These I put up here because this is your genetic code. This is how it's read. For every 3 nitrogenous bases on the DNA, we are going to then translate that into a very specific amino acid. The 3 sequence of nitrogenous bases on messenger RNA is called a codon. So I have a big long strand, messenger RNA, all these A's and U's and C's and G's in a specific order. For every 3, that's going to equal an amino acid. So if I have 3 sequence that is AGA, can everybody see that on there? What's the amino acid that's going to equate to in my protein? Arginine. If I have GGA, glycine. Okay? This is your code. Now I have to go read it. So that messenger RNA with all of its triplicate codons, remember it's a big long string of letters, is now going to go park itself on the ribosomes. And with the help of transfer RNA, we are going to now build ourselves a protein. This diagram here is going to be helpful to you. It's on page 106 and 107 in your textbook. And it gives us an idea of what goes on during transcription and translation. So DNA is the template. Messenger RNA is a copy of that information. Transcription. Messenger RNA then leaves the nucleus and goes and parks itself on a ribosome. And that strand of messenger RNA is systematically going to be fed through this little ticker tape machine, ribosome. There's 3 little sites there that we are going to read off of. The P site is where we dock with the other RNA that's going to help us build those proteins. That other RNA looks like, I think it looks like a bobby pin to me, I don't know. But it's also a sequence of nitrogenous bases bent in this particular way. At one end, you see another triplicate code. That's called the anticodon. At the other end of this particular transfer RNA, we're going to find a very specific what? Amino acid. This particular code is what we call the start codon. This is the beginning of any protein. It's called methionine. And we always start with methionine. That's going to get the whole reading process going. So, we have methionine, transfer RNA. What's going to happen next? Well, remember, these are enzyme-like things that are going to help us build new proteins. And what do enzymes do for me? They may break things down, but in this case, they're going to help put things together. So we have little places on that ribosomal complex where I'm going to allow things to come together. Particularly the things I'm talking about is the amino acid on the opposite end of that transfer RNA. So, in the P zone, I'm going to start my building of my protein. Free transfer RNAs floating around in the cytoplasm are going to be attracted to the next spot. They're going to come into place, and what's going to happen? Now, the amino acids that were on the opposite side of the transfer RNA are close enough to do what? Make a peptide bond. Yes? They're close enough together to undergo dehydration synthesis and link together. This whole unit is going to pick up and move over. What's going to happen to the transfer RNA that's empty? When it hits that E spot, it's going to get released, right? The amino acid it was carrying is going to be left behind in the P spot. What's going to happen next? Another one's going to come along. The same thing's going to happen. We're going to pick up, move over one. Everybody stays in the P spot. Whoever was in the E spot is going to do what? Get kicked off.
Now, when these are getting kicked off, is that the message? Is that the...
No, I'm reading the code. What's left behind? A chain of what? Amino acids. They're in that specific order based on what? The code.
Yeah, I don't get it at all.
You don't get it?
No.
I put these in this order based on this code. Where did I get this code from? DNA. Yes? These amino acids will be in that particular order because of my map. This messenger RNA read the map and now brings the information over so that it can be translated into a protein.
See, but what I don't get is when it releases the tRNA, there's no longer an amino acid connected.
What do you think is going to happen? It's chemistry. What do you think is going to happen? Once I release this, do I have free amino acids hanging around too? Yep. What's going to happen to this? It's probably going to attach itself to another amino acid.
There's nothing on it, correct?
There's no more amino acid on it, no.
Yeah.
It's still transfer RNA, still looking for that balance of chemistry.
But that amino acid that was on it is part of the peptide bond.
It's part of this chain now.
Yeah.
That chain is what?
Polypeptide.
Polypeptide. It's a protein. Now what does it do? Depends on the code, depends on what its job was.
So the next one that comes in into the A spot connects to the P.
No, it connects to,
Well,
these guys connect up here. That leucine connects to the proline that was on this P spot originally. This whole thing shifts over. Yes?
whatever protein's there.
Whatever amino acid was there is going to connect to the previous amino acid. Once this shifts, that amino acid chain is going to stay in the P spot, and whatever came to the E spot is going to get kicked out. Until when? I'm going to keep doing this. It's like a ticker tape. It's like a knitting machine. So I'm going to keep pulling it in and putting and adding on. I'm going to make my protein until I get it to the what? The termination code. There's a code that says stop. You're done. Once you get to the stop code, the whole chain's going to leave your protein and be processed. Once you get to the stop code, where are the stop codes? Up there, see them? So once in your chain of messenger RNA, once you hit UAA or UAG or UGA, you're done making your protein. That chain is then going to leave, and what do we do with the old chain of messenger RNA that we already read? Yep, we break it down and use it over. What do we do with the old transfer RNAs that used to be carrying an amino acid? We use them again, another amino acid, very specific amino acid, only to that shape, only to that anticodon is going to be attached to it. We'll start the process over again. So what we're left with, once we hit the termination code, is a protein. How that protein is going to act is going to be based on what? We already read the map. We already have the protein. What? Now it's going to do its job based on what? I'm going to react with something else based on my what? Shape. My shape is based on what? Aha! The order of amino acids in my protein. And who determined the order of amino acids in my protein? The code. Yes? So, let me recap. Transcription, translation. Genetic material in your nucleus, DNA, is a sequence of nitrogenous bases. That sequence is going to determine the order of amino acids I'm going to put together to make a protein. When I need to make a protein, a certain section of that genetic material is going to unzip. And I'm going to make a copy so that I can go make my protein. What molecule do I make a copy with? Messenger RNA. Messenger RNA is going to be formed in the nucleus. Where's it going to go next? That's transcription. Now I got to go translate it. Yes? Where's it going to go next? The secretary is going to leave the big boss's office and go type up the memo and send it out to the people. Messenger RNA is going to leave the nucleus, go park itself on a ribosome, and with the help of transfer RNA coming in and hooking up codon to anticodon, we are going to form a strand of amino acids. This order of amino acids is based on the code. Yes?
Now, when it's meeting up with these ribosomes,
Mm-hmm.
is that in the like the rough endoplasmic reticulum or the smooth?
Rough endoplasmic reticulum, yep. There are some ribosome and some protein stuff going on in free ribosomes.
What I'm asking was, where is this taking place? In there?
Yeah. So where's this chain going to end up? This chain of amino acids. It's going to end up in the endoplasmic reticulum, where we're going to kind of snip off. Anybody sew? You make curtains. You do some sewing. You put pieces together. You might have to snip some extra threads off here and there. And the processing of those proteins, the proteins that we've created from the code, is going to happen where? In the endoplasmic reticulum. Then those proteins are going to go and do their job. They're going to either be an enzyme that's going to help other reactions take place, or they're going to be a surface protein on your cells.
When you say that, is that inside the cell itself?
Yep.
You're talking about?
Yep. So when you make something in a factory, and that something is needed someplace else in the factory, what do you have to do with it? Transport it. So who's going to help me transport things within the cell? Endoplasmic reticulum, also diffusion. Some chemistry is going to happen to help push things from one place to another. So those proteins that we've created now are then going to go do their job. If it's an insulin, for example, in a cell, what am I going to do with that protein? It's in the pancreas. I made insulin. I need it throughout the whole body. What am I going to do with that protein now? How? How's it going to go into my bloodstream? And who's going to help me package and ship it? The Golgi apparatus, see? So it depends on what the protein is. Some of the proteins you make will stay within the cell because they're part of the cell's chemical structure. Some of the proteins will go sit on the plasma membrane and be part of the recognition system for the cell. But where do I get all of the information on how to make my proteins? From the genetic material. Does everybody understand how? What's the process of me making proteins called? Protein synthesis. This diagram is going to help you. Go through it. Look it over. All the steps are there. All the players are there. Remember the ribosomes. This is where I'm going to start to put things together. Now when we talk about proteins, we're not talking about, 20 different nitrogenous bases that make proteins. We're talking about thousands. So sometimes there's several ribosomes that are working on the same protein. Once I've made the protein, then it's going to be shipped and processed and put where it needs to go. So we see, ribosomal RNA. That's the ribosome itself. Yep. So the ribosome itself, the little docking station, is actually nucleic acid as well.
What makes the tRNA actually attract, to the messenger RNA? What makes
Enzymes. Yep. Remember those enzymes? There's actually enzymes that are going to help the whole reaction take place too. And again, once I'm close together to react with another amino acid beside me to create a peptide bond because I'm in that slot, that reaction can take place as well. So sometimes I need to package and ship. The endoplasmic reticulum is the place where I process protein, and then somebody else might take over for me, like the Golgi apparatus. So what I see is from DNA sequence to protein, I see the sequence, the triplicate code on DNA that gives us a codon messenger RNA that's then read or attached to by the transfer RNA. And look at the original triplicate code in the DNA. It's exactly the same as what's on the transfer RNA, isn't it? Except uracil in place of thymine.
So the codons, are those what was on that chart, right? Okay.
Yeah, and there's different books have different charts. Sometimes that chart is of your anticodons, sometimes it's of your codons. This particular one is codons. So basically, if I look on the chart, I'm going to see the sequence, and let's go check it. You go check it. If I see the sequence UCG, it's going to say serine. Go check it. Does it? Yep. So sometimes, depending on the textbook, it's written in either way. But that particular triplicate codon is always going to be a very specific amino acid. Yes? You understand now how DNA is going to dictate what proteins you make. It's based on the series of nitrogenous bases. And about 2%, probably less, of your genetic material is different from mine. That's how many nitrogenous bases we're talking about. So there's billions of these that help us make our proteins. Again, many of our proteins are very similar, but many of them, enough of them, I should say, are different, and they make us who we are as separate organisms. Okay? So that's transcription and translation, and that is protein synthesis. Any questions? You're going to get 100 on your exam. That's the stuff I trim out later. Okay? So introns and extrons, when you talk about genetics and specifics, they're referring to different portions of the code. Not all the code is useful. All the code is needed to line everybody up and to create that whole process that we talked about. But some of the stuff is extras. And that's what introns and extrons are. Extrons are the good stuff, and introns is the extras. When you create new cells, as your cells age and they create new genetic material, some of the ends are trimmed off. Every time I replicate DNA, some of the ends, the extras, are trimmed off. Those guys, and you don't have to write this down. Those guys are called telomeres. As your cell ages, as it replicates more and more and more, those telomeres become less and less and less, to the point where we get to a point where there's no extras anymore. And what's going to happen to that cell? Is it going to be able to replicate anymore? No. So as we age, we see our cells will change a little bit. Another thing we're going to see in our cells, not to scare the bejesus out of you or anything, but other organisms like to incorporate their DNA into your DNA. Do you know what those organisms are called? Viruses. As you age, there's a certain percent of your code, your map, that is not yours. It's viral. Scary, isn't it? And how do viruses replicate themselves? Same process, except who carries it out? You do. So you do this for them, so they can make more of themselves.
So what do we get out of it?
Not much. There is no benefit. They take over our machinery. So when you get sick, say you get the chicken pox, the virus takes over your cellular mechanisms to make more of itself. Now, depending on the virus, it can kill your cell. It can wreak havoc through your system. But sometimes it can be smart and just lay low and just hang out with yourself for many years. One of the things you're hearing a lot about now is something called shingles. It's the same virus, right? That's been hanging out, just hanging with you for long periods of time until something sets it off. Stressors. And what does it do? It starts to replicate. It takes over your mechanisms and starts to replicate itself using your mechanisms again. It manifests itself in a different way. Not too shabby. Very smart little viruses. Yes?
I know if you take antivirals very quickly, it can help. What do the antivirals do?
basically your cells, proteins on the surface of your cells, there are special proteins on the surface of your cells that viruses actually use as doors in. Okay, and they need those proteins to get into your cells so that it can take over your replication mechanisms. What antivirals do is try to block those entryways. Your cells actually do that for other cells. And we'll talk about that next semester when we talk about the immune system. They produce substances called interferons that will protect other cells from becoming infected by that virus. Okay, and that's what a lot of the antivirals are doing. It's not going to make you better, but it's going to prevent more cells from becoming infected with that particular virus, so that your body can then get rid of that virus. What cures a virus? Your immune system. So every time you're sick, there's no need for you to always take antibiotics, right? Antibiotics kill who? Bacteria. Most of us aren't sick from bacteria. Most of us are sick most of the time from who? Viruses. And who's going to cure a virus? We are.
They don't go away, though.
Yeah, it depends on the virus, though. Depends on the virus. Again, we'll talk about that next semester. All right. So we need to understand the membrane transport system, all of the organelles, endocytosis, exocytosis, the structure of the plasma membrane, phospholipid bilayer, all those wonderful little organelles.
Now, on the bilayer, the little head part, the little round part, that's the polar side, correct?
It's the hydrophilic side. The middle is the hydrophobic.
Yeah, but which
It's
side?
polar because it's got two opposite sides.
Well, in the mastering AP, it's asking you specifically which side of that is polar, and it points to it. So, because if it's because it repels. So what is below it keeps the cytoplasm and everything into the cell, and what's on the outside keeps everything from coming in. So does that makes it polar, correct?
Correct. It's got two opposite sides. That's what polar means. So it also has opposite charges. So on the outside of the membrane, we have a positive charge versus the inside of the membrane with a negative charge. Yes?
Yeah. That's all I wanted to know. I mean,
Yep. Good. Hopefully I answered your question. So remember, understand it's not only the phospholipids that make up the membranes. What else is there? All those proteins, all those carbohydrates attached to proteins, all those carbohydrates attached to lipids, glycolipids, glycoproteins are part of the plasma membrane as well.
On mastering AMP, it talked about integral proteins
Yep.
and identifier tag. And in the book, it shows one of the integral proteins with the glyco something or other off of it as
Yep.
an identifier.
Yep.
But the identifier was then the answer that we were supposed to come up with was an identifier was just the little glyco whatever
Right.
on top.
Correct.
Okay, so the book, it's not actually the protein itself.
Well, the protein with the carbohydrate attached to it.
No, when you say identifier, you mean tag?
Yeah, the tag. The tag is the carbohydrate attached to the protein.
Yeah, and I put that on there and I got it wrong.
All right, don't yell at me. Talk to your lab teacher. Questions on mastering are lab questions. And what you should do if you have a problem with a question, because these are canned questions from Pearson, okay? I'll tell you right out. Instructors will go and pull out questions that they think are pertinent to the lab material. If you have a question on any of those questions, bring it to lab. talk about it with your instructor. Maybe it's a crappy question and we need to get rid of it. Your instructor's not going to know that until you come and talk to them about it. And they can alter it or get rid of it. Okay? So don't get mad at me. Yes? What do we need to know for Thursday? Everything we've discussed in class is everything you need to know on Thursday. Do I give you a study guide? No. Study everything. Same format for this test, not for all tests. We have essay questions on in the future. I give you a break on the first exam, and the only thing you have is multiple choice.
How many questions?
100, at least. Okay?
Are we spending all of Thursday just doing tests?
Correct. No pressure on Thursday. You come in, you take your exam, you go. I'm not going to, no. It's open mind. Okay? In that picture, I know that sometimes protein synthesis can seem overwhelming because in your textbook, we talk a lot, it talks a lot about all the specifics, okay? But look at that diagram that ties it all together, because sometimes if you look at a picture when you try to understand a concept, it makes more sense. Okay? So pull out your clickers. It's time for a clicker question. If there's anything specific you need answered, ask. This is your time. Let's do a couple of clicker questions, and then I'll ask you that question again. So, the basic function or functional unit of living organisms is your answers you're clicking. Five, 4, 3, 2, 1. Only 55? What's the answer? Cells. C, 4. Ah, good. The three main components of all cells include the plasma membrane, the nucleus, and the all, I'm going to give you a hint, all cells. That's your hint. Five, 4, 3. What's the answer? Thank you. All cells have cytoplasm. Do all cells have DNA? No. Red blood cells don't, right? Remember I told you about the exception? Organelles. Yep, it's part of the cytoplasm. Cell walls. Who has cell walls? Plants. Phospholipids orient themselves in aqueous solutions such that and this would help you with the answer to that question on mastering. Five, 4, 3, 2. Well, hurry up and stop talking. right. What's the answer? The answer is D.
Why weren't these on the test? These are all the ones I know.
A cell might increase its membrane fluidity or flexibility by increasing the amount of what in the cell membrane? Five, 4, 3, 2. Ooh, I got a 56. What's the answer?
So explain that then.
So the more unsaturated, and I would never ask you this question, but the more unsaturated fatty acids you have, if you remember what a phospholipid looks like, you have a phosphate and 2 fatty acids. The more unsaturated a fatty acid is, the more bends there are in it. The more bends there are in it, the less rigid it is. So it's like putting a hole in a wall almost. So I can allow things to go through it, have it be more fluid if I have more bends in it. Then I wouldn't ask you that question, so don't worry about it. In areas of the body subject to a higher degree of mechanical stress, which of the following types of membrane junctions would you expect to be most prevalent? Pulling on those cells. If I pull on them, who's going to help? Wait a minute, let's finish the question. 5, 4, 3, 2. What's the answer? Desmosomes. Spot welds. Remember I described them as spot welds? So if I'm making a building and I want to keep beams together, I put spot welds on them to hold the pieces of metal together. Hold them tightly together. Microvilli is going to allow things to spread over the top of me. Tight junctions are just going to hold me together. And gap junctions are like little straws that poke through the membranes, and they're going to allow things to pass from one cell to the next. So it's those desmosomes that are going to hold me tight together. Question?
Well, actually, no, I'm getting to it. So a tight junction is like a weaker...
It's not weaker, it's just the regular connection. If I want to enforce that connection, I'm going to throw some desmosomes in there to make it even.
All right. Now, microvilli are finger-like. They're just finger-like.
They're like hair. They're like hairs.
Yeah. So, and cilia are also very similar, correct?
Correct.
That are finger-like.
Correct.
Now,
Cilia and microvilli are similar.
anyways, but what is the difference between the two?
One's bigger than the other.
But they both...
Microvilli are more for increasing surface area. Cilia are longer, and they're going to help move things across the surface of that cell group.
Because they both...
They're made of the same structure, and they basically kind of do the same thing, except one's longer than the other.
The question in the mastering was finger-like projections that are used that have large surface areas for absorption. believe is.
And that's going to be microvilli.
And actually cilia, it sounds like finger-like.
Cilia aren't typically used by the cell for absorptive purposes. They're used for movement purposes. So cilia and flagella are more for movement. Microvilli are absorption, increased surface area. Good? Yes, sir.
Yeah, we're going to need to know the field terms, like the very specific ones.
Yes.
So all of them.
Yes.
Are we going to have
Yes.
diagrams on our test?
No. No diagrams on your test. But similar to the quiz, I'll ask you in question form about your regional terms. If I am facing you, looking at your front, is that anterior or posterior? That's the way the question will be couched.
I was thinking more like the mantis.
Yep, all of them. So is there a lot of material? Yes. When should you have started studying? Notice that's past tense. When did we start class? Yeah, that's when you should start studying. So, seriously, you should be studying all the time, all of this stuff. See you on Thursday. The only way you will have an excuse for not taking the exam is if it's a written one. Be here.